Hello, my name is Jaelle Brealey and I'm a postdoc at NTNU University Museum. And today I'm talking about work I did in my previous postdoc at Uppsala University in Sweden with Katarina Guszczanski. And in this study, we used oral microbiomes from museum specimens of wild animals, particularly the Scandinavian brown bear, to study the history of human antibiotics use. And these different aspects might seem very unconnected, but I hopefully will convince you that it does make a lot of sense as we go through. So let's start with antibiotics. Antibiotics were discovered by humans in the 1930s and 40s. And since then, we've discovered various different classes of antibiotics. However, since the first mass production of antibiotics, we very quickly discovered antibiotic resistance develops. And this has happened to each of the different classes through history. So antimicrobial resistance is emerging as a global health threat. And these statistics are a little old, but in Europe, for example, in 2015, it was estimated that around 30,000 deaths were attributable to resistant infections. And globally, by 2050, it's estimated that there will be around 10 million deaths due to resistant infections. Resistance, well, bacteria can be multi-drug resistant, so they have resistance to multiple different antibiotics. And in some cases, they are extremely multi-drug resistant to the point where they are resistant to all known antibiotic treatments. So it is a really big problem. And what makes things more complicated is that antimicrobial resistance, or AMR, elements are easily transferred between bacterial species, even for quite unrelated taxa. And this transfer can occur through plasmids, bacteriophages, recombination, and things like that. This easy transfer of AMR elements means that it's quite easy for resistance to spill over into wild environments. So for example, when humans use antibiotics in medical settings or in agriculture, for example, in feeds as growth promoters or in domesticated animals as a veterinary treatment, resistance can develop and be shared between humans and animals. However, this resistance and antibiotics can then also spill over into wild environments, for example, into the soil and water sources from human sewage and animal waste. These wild environments also include wildlife. And it is then possible that wildlife could act as reservoirs that would then allow resistance to get back into human and domesticated animal populations. What is still unclear is to what extent this happens and the extent to which wildlife can act as reservoirs for antimicrobial resistance. This problem is further complicated by the fact that AMR is actually a normal function of natural environments. So these normal functions include, for example, tolerance of bacteria to different toxins like heavy metals, competition between bacteria trying to colonize the same niche, and evasion from host defenses like fungal or plant defenses. And resistance has been found in across the globe, so even in the most pristine environments such as the Arctic or Antarctic. This makes it difficult to study how any microbial resistance has spilled over into the environment as a result of human use. Resistance has been found in ancient environments, so in the pre-antibiotics era. So for example, in permafrost sediments and in medieval human oral microbiomes, so in the dental calculus on the teeth of these medieval specimens. Since this shows that we can find antimicrobial resistance elements in historical and ancient samples, we then decided to look at resistance in the oral microbiomes of wild animals from historical museum specimens. And we're doing this using a substrate called dental calculus. So dental calculus is the calcification of the oral, the microbial biofilm that forms on vertebrate teeth. And this biofilm, which is part of dental plaque, it includes the components of the oral microbiome, as well as host DNA and food debris. Then periodically over an individual's life, this plaque calcifies into dental calculus, as seen here. And then a new biofilm forms on top of the calcified dental calculus, and then this just keeps happening so that it layers. And then after the individual's death, this calcium matrix then preserves the DNA within, and we can characterize this with DNA sequencing. This means that museum collections are really great resources for dental calculus and in sort of insight into past oral microbiomes. Now, we're using Swedish brown bears for this, which might not seem intuitive, but I hope to convince you that actually Swedish brown bears are a really good system for our study. They are widely distributed across Sweden, particularly central and northern Sweden. And as omnivores, they have they do not have a specialized diet, and they aren't found in close association with human settlements, which means that the AMR, that if we find AMR, then it's not as a direct result from sort of direct interactions with humans or human waste. More practically, we have access to an extensive museum collection of brown bear skulls from the Swedish the Swedish Museum of Natural History. And they have a collection spanning the last 150 years. And you can see an example of bear dental calculus here. Finally, we also know from previous studies that the brown bear oral cavity in some ways does resemble humans. So they do suffer from dental diseases like humans. And we know from our own previous study of brown bear dental calculus that they also do have some component of their dental calculus microbiome that is similar to humans. So here we have a kind of classic source tracker plot, which is very popular in ancient metagenomics, where each bar is a different bear calculus sample. And then on the y-axis is the proportion of the calculus microbiome, which is similar to a different set of reference sources. And I just want to focus on the modern human dental plaque and modern, historical human dental calculus in the yellow and orange. And you can see that a lot of the bear samples do have a significant proportion of their oral microbiome, which is recognizable as oral based on human sources. We've also found a number of known oral bacteria in bear dental calculus, including karyogenic bacteria like lactobacillus, various streptococci, as well as red complex bacteria, which are present in healthy humans but are also associated with periodontitis. So this means that we can use our current reference databases to identify oral bacteria in the bear oral cavity. Finally, Sweden itself is also an interesting case study to look at AMR because, like the rest of the world, the pre-antibiotics era, so before sort of 1940, is the same. And then in around the 1950s, they started mass producing antibiotics. And then in the following 50 years, rising resistance did occur in the population, in the human populations, but they were one of the first countries to start trying to combat this resistance. So for example, in 1985, they banned the use of antibiotics in animal feed as growth promoters. Then in 1995, they introduced the Strategic Program Against Antibiotic Resistance, so STRAMA. And this program includes monitoring for antibiotic resistance. It also they also regulate the sale of antibiotics and track that over time. And so based on all this, our key questions were, our key aims was to establish the baseline of AMR load in the Swedish brown bears in the pre-antibiotics era. We then investigated whether AMR load correlated with human antibiotics use in Sweden over time, so through this period of rising resistance in the human population. And we determined whether the Swedish public health policies, whether they were effective at reducing spillover in the 2000s. So like I said, we did this using museum specimens. So we first had to collect dental calculus from the teeth of brown bears, and then we did DNA extractions and library builds in our ancient DNA lab. And you can see my students Tice and Henrik hard at work here. We followed this with shotgun metagenomics DNA sequencing on an Illumina platform, followed by data quality control and processing. And I won't go through all of those details today, but I do want to focus on one important part of the quality control, which is dealing with contamination. So contamination is a known problem in any ancient or historical sequencing study, particularly in metagenomics. And it can be introduced to the sample through the environment where the specimen was found, storage in, for example, museums, at any point of handling during the storage, as well as during laboratory processing. And we have a number of strategies to deal with contamination. This includes processing all of our samples in a dedicated ancient DNA facility with full PPE. We also decontaminate the surface of our dental calculus samples before DNA extraction using UV irradiation and an EDTA wash. We collected negative controls both in the museum environment when we were sampling, as well as including extraction and library blank controls during laboratory processing. And then we have a number of bioinformatic methods to try to identify and filter out contaminants, including the one I'm showing here, which is a R package called Decontam. Finally, for this study to be extra sure that we were getting oral taxa and not environmental resistance from environmental bacteria, we subsetted our data to only include known members of the oral microbiome. And we had to base this list on human and domestic modern oral microbiome studies. This was because we don't actually have any modern brown bear oral microbiome studies. But what this also means is that we're really focusing on bacteria that are carrying resistance elements that could then actually colonize humans, and so therefore are of medical relevance. All right, so then the last step was to identify AMR in our sequence reads. And we use the Comprehensive Antibiotic Resistance Database, or CARD database, for this. And this database is curated and it's annotated. And this allowed us, using BLAST, to both quantify the total AMR load in our samples, as well as assign resistance gene families to our reads, so that we were able to actually look at some of the different functions that were happening in the oral microbiome. So to start off with total AMR load, which is the number of reads that map to the CARD database for each sample, divided by the total number of oral reads in that sample. And focusing first on the pre-antibiotics era, you can see that from our samples from before the 1950s, there is a natural a baseline of natural AMR present in the brown bear dental calculus. Then moving on to look at the following 50 years, we can see that resistance does increase in our specimens, and this correlates with increasing antibiotics use and increasing resistance in the human population. Finally, turning to post-2000, we can see that resistance in our brown bear specimens actually does decrease, which is in line with the introduction of Swedish public health policy control measures. And this quadratic relationship, where it kind of starts lower, increases during the 70s and 80s, and decreases again, was significant. So this shows that the total AMR load in our brown bears does reflect human antibiotics use. We then looked at the diversity of antimicrobial resistance genes, or ARGs. And so we firstly just counted the number of different ARG families that were present in each of our 5 time periods. And you can see that particularly in the last 2 time periods, so from 1986 onwards, we see a increase in the number of different ARG families present. When we look at what these different ARG families are, which you can see in the heat map here along the bottom, we can see that some ARG families are present at consistent levels, are consistently present throughout the 5 time periods. And in this heat map, red indicates higher abundance of the ARG family, and blue indicates a lower abundance of the ARG family, and gray are ARG families that were not detected in that time period. And you can see that we have a number of ARG families that were only detected in the last 2 time periods, but at low abundance. And most of these ARG families have mechanisms associated with antibiotic inactivation. And many of them are from ARG families or classes of resistance genes that have expanded in the last 20 or 30 years due to increased pressure from humans using antibiotics. So for example, OXA is confers resistance to beta-lactam antibiotics like penicillin. So we next looked at the overall structure of the oral microbiome. So we're seeing these changes in antimicrobial resistance over time. But when we actually look at the composition of the oral microbiome in the bears, we don't see any great shifts over time. So in this ordination, each point is a sample representing the entire oral microbiome for that sample, and they're colored by time period. And then on the right-hand side, this is the exact same ordination, but each sample is now colored by total AMR load. And you can see that there's no clustering by time period or total AMR load. There's no structure to the plots. So this tells us that the changes we're seeing in AMR load is not associated with a big change in community structure. Instead, we found that specific ARG family abundances were correlated with specific bacterial genera that colonize the oral cavity. And one example I'm showing here is from the RNA polymerase, particularly the beta subunit, which is shown in red in this protein structure. And this family undergoes mutations in this subunit, which confers resistance to antibiotics related to rifamycin. And the ARG family load over time mimics the total of change in AMR over time, so that we see lower levels pre-1950s and then increases in the 70s and 80s, followed by a decrease post-2000. And the abundance of the Robi family was correlated with the abundance of the oral bacteria Actinomyces. And you can see this in the correlation plot here, as well as if we look at the abundance of Actinomyces over time, and we can see it also mirrors the changes over time in Robi. To take a different example, here this is a efflux pump, so the R&D efflux pump, and we see a strong correlation between the abundance of R&D and the abundance of Neisseria. And Neisseria have an efflux pump called MTR-CDE, and you can see the structure here. So this sits in the membrane of the bacteria, and it has multiple different roles. So as well as pumping antibiotics out of the bacterial cell, it can also remove toxins, hormones, and other peptides. So it has multiple different roles for the bacterial survival, not just resistance. And we see that this this R&D pump increases in abundance over time. And we see that the abundance of Neisseria also increases over time in the same manner, showing that the changes we're seeing in global AMR seems to be driven by these specific changes in the community and not an overall change. Finally, we also looked at the bear sampling locations and whether they were found in areas of a lot of human activity to see whether or not these bears were likely acquiring this resistance from close interactions with humans or human waste. So on the map of Sweden here, we have each of the sampling locations of our bears colored by time period. And you can see that we have a good distribution across Sweden without sort of any tight clustering by a certain time period. And we have a number of samples where we don't know exactly where they were located, but we know that they were from like the county. And underlaid on the map is the human footprint index. And this is a measure that was taken in 2009, which kind of is a single number to represent human activity in that area. So it includes data on land use, human population, transport networks, and other human infrastructure. And the darker the color, the higher the index. So the more human impact is on that location. And we found no correlation between the human footprint index and the total AMR load. However, you can see from the map that most of our bears were actually sampled from locations that were low in human activity, so the lighter parts of the map. And I've plotted the Swedish average for 2009 just to emphasize that most of our bears are coming from regions that really were not close to human settlements. Of course, this data was from 2009, and most of our samples are from much earlier than that. And we don't really have good consistent data from the last 200 years of land use, but we do have really great records of human population density from parish records of Sweden, which go back hundreds of years. So then we also looked at the historical human population density for each of our sampling locations at the time period in which it was sampled. And again, we found no correlation between human population density and total AMR load. And again, most of our bear specimens are from locations with a density much lower than the Swedish average. So this suggests that our bears are not acquiring AMR from close interactions with humans. So to summarize our results, if I go back to this schematic of the spillover of AMR into the environment, we established that there is a baseline of natural AMR in our pre-antibiotic era bear samples. We also found that the AMR load in bears correlated with human antibiotics use in Sweden. So it increased over time until 2000. However, we found that it was not, the AMR was not associated with the bear proximity to human environments, which suggests that we're seeing a widespread spillover of AMR into the environment, including into wildlife. However, there is a silver lining. We also found that there was a decrease post-2000, which suggests that Swedish health policies were not just effective in limiting resistance in human populations, but were also helping limit spillover of AMR into the environment. And it would be really interesting to replicate this study in other regions that had different policies to Sweden, or maybe introduced control policies at different times to see if we see the same results. So with that, I'd like to acknowledge all of the co-authors on this paper. So Katja Koscianski, my fabulous PI from Uppsala, and my fantastic master students, Henrik and Thijs, as well as our museum curator collaborator, Daniela from the Swedish Museum of Natural History, as well as everyone else in the Koscianski lab while I was there. I'd also like to acknowledge the amazing publicly available data sources from Sweden. So the population data came, or the historical population data came from SIDA, and the modern population data from Statistics Sweden, and our land use data came from Lantmäteriet. Then, of course, I'd like to acknowledge our funding sources and computational resources. And if you want to read more about our study, you can see our preprint, which is available on bioRxiv. And since this is an online seminar, if you have any questions, feel free to get in contact with me on Twitter. And thank you for your attention.
Yay! I'm gonna do the applause for everyone who's listening. That was very cool, Jaelle. Thank you so much. All right, I don't know. Do you want to stop sharing maybe so we can see each other better? I suppose.
We can do that. Yes.
Perfect. Fantastic. So look, you know what I was wondering? So really, really cool, I think. But how the hell did you get started on that? So, where is this entire idea coming from? And did kind of your interest before prepare you for these crazy excursions?
Yeah, that's a really great question. It's one of those where you're like, where did the idea come from? I've always had my background is in infectious disease. So I've always had a interest in particularly human infectious diseases. So that includes antibiotic resistance. Um, and then we were already working on oral microbiome evolution in bears and other Nordic species. And then when I read the paper by Tina Warriner, where they found the AMR in the medieval human oral microbiome, human dental calculus, it seemed like something that would be really cool to see. Okay, do we see this in any of our brown bear specimens? And then when we did in our pilot study that we published in MB early last year, then when we had this like great resource at the NRM museum that would really span this time frame, it seemed like a really cool thing to look at.
Yeah, sounds good. And do you think? I mean, I'm just trying to think this through. I mean, are bears indeed the best study system to look at the spillover of human antibiotics and resistance factors into the environment? So say, if not for this great resource that was available, if you had to design the study from scratch, would you have chosen something else to look at?
Yeah, I mean, I do think bears are quite useful because they are not closely associated with humans, but I think it would be really interesting to do a comparative study where you take different animal species, some which have, like bears, have very little association with humans, and some which are tightly associated, like rodents, and also looking at the different ecology of the animals. So like badgers, for example, which spend a lot of time digging in the ground, might be more likely to acquire soil-based microbes. So I think actually the comparative point of view would be really, really interesting there. And seeing how the animal ecology affects their likelihood to to acquire human-associated AMR would be really valuable. But I mean, of course, we are also limited by what we actually have, what museum specimens we have available.
Yeah, and I guess from that you made a very good point that this transition through time is extremely important, so that one can see the rate of change through time compared to the baseline that is expected not to be affected by humans, right?
Yep, exactly. So we really need specimens that actually cover those key time periods.
Yeah, cool. But then, kind of getting a little bit further from what you just said, how? So you mentioned these different species that maybe have different ecologies or different levels of exposure to human environments. But how general are the results of your study? Like, if one looks at, I don't know, the effect of policies or maybe the effects of human antibiotic use. I mean, can you comment on that?
Yeah, I guess, I mean, this is just one study in one case study in one region. So it would be really interesting to run the similar study in different regions to look really at the effect and see how general it is. So I think this has great potential to be used, like this idea of using dental calculus to actually look at the temporal changes. Because we know so many different animals do have dental calculus, so we could look at different regions and really have an idea of do policies make a difference? Is this just like a one-off case in Sweden, or can it be applied more generally? I think that is actually a really interesting question.
That's right. I mean, it would be fantastic if that could be confirmed, because it would basically suggest that we as humans can mess up so many things in the environment, but at the same time, we can turn back the wheel in a sense, right? And
Yeah, yeah, exactly. And I think there's so much focus on how much we've screwed up the planet, which is absolutely true. But it's always nice to actually see, okay, our actions can make a difference. And maybe if we do bring in these policies, we could, yeah, as you say, like turn back time and make things better.
yeah. All right. So if you were me, what type of question would you ask of yourself? Let me formulate differently. Is it something that you would want to share that you didn't have the chance to talk about in the seminar?
I mean, not really. I mean, I guess just highlighting how great dental calculus is and how useful it is. That's always my, yeah, I've talked about this before, that it's this amazing resource. So, you know, we're using it here to look at antimicrobial resistance, but we've also, you know, used it to try and get host DNA, to look at host genomics, to try and get reconstruct diet, to look at oral pathogen evolution, oral microbiome evolution. So yeah, it's a really cool material.
So yay to crazy samples, is it?
Yeah, exactly, exactly. It's the value of looking into weird things that people think, no, there's nothing useful in this, and actually making the most of that. And these sorts of, yeah, exploratory studies.
Isn't that what the alchemists were trying to do, turning lead into gold?
Yeah, exactly. That's what metagenomicists are, just a modern day alchemist.
Well, maybe that's the perfect point to stop the seminar. On this type of statement. That was very cool, Jaelle. Thank you so much for sharing your research.
No, thank you. Thank you for the invitation. It's been great.
Okay, thanks. Bye.
Bye.